we will talk about the article from New England Journal of Medicine. The article that is Rhinovirus Wheezing illness and genetic factor of childhood-onset asthma. First, we will talk about why is the problem important. From the other research, we know that genetic variation at 17q21 locus is associated with asthma. The effect is longer in children who has been exposed to environmental tobacco smoke and viral infection. From this pie graph, 80% of asthma is caused by viral infection, and 2/3 of viral infection is HRV or human rhinoviral infection. And infancy who have HRV infection with wheezing illness is significantly increased risk of asthma. However, exposure to HRV infection doesn't lead to wheezing illness in all children, and wheezing illness doesn't result in asthma in all children too. So, this research is a study about the association of genetic variation, viral infection, and asthma.
Contributive of this research is that the first genome-wide association study of childhood-onset asthma revealed a susceptibility locus on chromosome 17q21.
There were many previous researches that show that 5 single nuclear polymorphisms on the chromosome 17q21 viral respiratory infections and environment are risk factors of childhood onset asthma. However, there's never been any study that elucidates the relationships and influence between these factors. So the differences of this research are: 1. They hypothesize that genetic and environmental factors can affect each other in either additive or interactive manner. And 2. They use an in vitro model of peripheral blood mononuclear cells to find HRV responsive genes and to identify the relations between viral infections and genetics.
There are some limitations in our study. First, because HRV is the most common virus encountered, we cannot exclude the possibility that the absence of an observed with interaction with RSV due to insufficient power. We analyze viral exposure in children with wheezing illness, and we cannot establish causality. Maybe HRV wheezing illness is merely marker of the underlying predisposition for asthma, and is not causal. Finally, we were unable in this study to evaluate the role of the concomitant bacteria, which also trigger episode of wheezing.
For the method, this research used 3 groups of cohort study. So the cohort study means a type of medical research used to investigate the cause of disease and to establish link between risk factor and health outcome. So the first group is the childhood origin of asthma or COAS. Second is the Copenhagen prospective study on asthma in childhood or COPSAC. And the last is a group of adult volunteers. For COAS, we recruit 289 newborn which enroll in medicine Wisconsin between November 1998 and May 2000. For COPSAC, we recruit 411 children which enroll in Copenhagen between August 1998 and December 2001. For volunteer, we recruit 100 unrelated adult volunteers in Chicago between July and November 2011. For next step, we obtain blood sample for study genotyping SNP. And we use the previous study we found 5 asthma association 17q21 SNP to use in test. For the test, all 5 SNP test in co-as, but COPSAC and adult volunteer, we use only SNP RS7216389 because it's nearly perfect linkage disequilibrium.
In the previous work, we have seen that there's a very strong association between asthma and 17q21 variations. There's many genes in these variants, but we interested in 3 genes as above. And the results of the study shows that in RSV infection patients, there's no relationships between these genes. But there are significant upregulations of GSDMB and ORMDL3 in HRV infection patients. And GSDMB have very unclear function, but there's a work suggests that ORMDL3 overexpression they may increase in efficacy of the HRV infection and HRV replication in respiratory epithelial cells and also decrease in respiratory epithelial cell reparation ability. So we may say that having a HRV infection may increase risk in wheezing illness in childhood in a child who contain 17q21 variations. Thank you.